worth. And so we have a little bit less of brand. Let me see what. Here we go. A little less of brand new information. So these are the three components of glucose digestion. All right?
Is there a page that should be on the left?
Yeah, hold on just a second. I think it's 50. Sorry. Wrong group. Are you there from that table right there? Page 56. Now, glucose is the most prominent fuel used by the body. If glucose is not available, as in diabetes, where insulin is either the cells are resistant to insulin or the pancreas is not producing sufficient insulin to enable the cells to obtain the glucose, the cells will resort to additional fuel sources. They'll use proteins, they'll use amino acids as a supplement. So on Tuesday, when we finish this up, before we move into the hormones, we'll look at how proteins or fatty acids and glycerol can enter into this cycle. So glycolysis, the current term is citric acid cycle, and electron transport are the three stages in which glucose is broken down and all of the energy available is captured and stored as the bond between the third phosphate in ATP. So total amount of ATP available is about 38 molecules of ATP from 1 molecule of glucose. That energy comes from the energy found in the hydrogens and the electron of each hydrogen predominantly. And most of that happens at this point. Glycolysis is the only component of these stages that does not require oxygen. The series of enzymatic reactions in glycolysis is anaerobic. The entire glycolytic sequence can take place even if the cell doesn't have oxygen. However, oxygen is required as the very last step in electron transport. If that isn't there, then these steps back up, citric acid cycle backs up, and the last product of glycolysis, pyruvic acid, is converted to lactic acid. So these two steps, which take place in the mitochondria, remember the mitochondria is the end delivery point for oxygen. It's our respiratory organelle. And these are oxygen requiring or aerobic. Okay. Now glycolysis takes place in the cytoplasm. So without being membrane bound, except for the membrane of the cell. So just a brief review for cell organelles. If the enzymes that regulate glycolytic activity would be produced using what structures of the cell, as opposed to the enzymes regulating citric acid cycle and electron transport? How would those proteins be made? Free ribosomes, exactly right. Okay, because they're not ever surrounded by a plasma membrane. So these enzymes are synthesized solely by the interaction between messenger RNA and free ribosomes. Because these enzymes take place in the mitochondria, they have to be directed to a location. And whenever that happens, if the location is the lysosome or the mitochondria, or if they're antibodies and they're being secreted out of the cell, then the ribosomes interact with and messenger RNA are bound to ribosomal ER, and then the new proteins are sent to the Golgi apparatus for modification. But of course, that is a more efficient way of doing the enzymatic reactions because those enzymes are confined to the mitochondria and it's not a random interaction between the molecules and the enzyme. All right, so that makes it a more efficient mechanism. So we're just going to talk about glycolysis. All right, and you'll notice on page 56 of your lecture notes that underneath glycolysis, kind of in the middle of the page, is a series of statements labeled summary. So for each of these components, that summary is what you need to know. You'll find a lot more pages in your textbook, but I focused it for you. And the summary for glycolysis, the summary for the intermediate step, summary for citric acid cycle, electron transport, is what you need to know from these components. Now when we look at glycolysis itself, one thing I want to reassure you, you're welcome to still learn it, but you do not have to memorize the enzymes for each of these steps. I do want you to know that they're enzyme specific, just like our breakdown of polysaccharides. If the right enzyme isn't there, that reaction doesn't take place, okay? So you may have had to learn these for micro. I know my son in high school had to memorize them, but I've forgotten them and I'm not going to ask you to learn them. So we have 3 different sugars, all of which have 6 carbons, all right? So galactose, glucose, and fructose all have 6 carbons and can be utilized in here. In fact, we take glucose and change the shape of it and turn it into fructose. So let's look at the steps that are involved. We take a 6 carbon, and I'm not going to draw the ring shape, nor put in the oxygens and hydrogen. So oxygen and hydrogen are present here, okay? I'm just putting in the carbons of the molecule, and this is glucose. To get this process started, an activation energy is required. You've probably heard about that in chemistry. We have the potential energy in the molecule, but it's not going to start the reaction until we actually push it, like getting a boulder to go over a little lip before it actually falls down the cliff and crashes into the VW bug. And so the activation energy for this process requires 2 molecules of ATP. So 1 molecule of ATP provides the energy and the phosphate group to put a glucose on 1 carbon. A phosphate group on 1 carbon. This enzyme that regulates this reaction is irreversible. So once this reaction starts to take place in a cell, that we can go backwards up to this point. So we can take any of our products and recombine them back to glucose. We can't take this last phosphate off except in the liver. All right? And that's important in the liver because we're storing it as starch and breaking it back down to glucose and storing it as starch. And so that's necessary that this step be reversible in the liver. Okay? Then we repeat this process and we add a phosphate group. We use the energy of an ATP molecule to add a phosphate group to the other carbon. And we modify the bonds from a 6-sided ring, which is glucose, to a 5-sided ring, which is fructose. And so, well, let me change this here, plus ATP. So now we have our 6 carbons of the phosphate group. And so this is now known as fructose 1,6-diphosphate. But it doesn't have any different atoms than glucose does. It's just arranged slightly differently. Notice we've started our glycolytic reaction. We've used up 2 molecules of ATP. We've added to our glucose molecule instead of breaking it down. Okay? Now we're going to start to break it down. Again, a specific enzyme will work just on this molecule. Fructose can come in here directly. All right, the ATP can bind directly to fructose. for the first time, we break a carbon-carbon bond. Note that no energy is going to be released more than a couple of ATPs. And that comes from the hydrogen components. So now, this is actually reversible. We have 2 molecules of a 3-carbon component. Sufficient energy was released, and that is enough to bind a phosphate group to 4 molecules of ATP. Yes?
Between the 2 and the P instead of 3, is that a reversible arrow?
Yeah. I started to draw one arrow, then made it. Forgotten all of a sudden how to draw it here. There we go. My brain is not allowing my hand to work. So this 3-carbon molecule now, remember what we called the 3-carbon component of our triglyceride? Glycerol. So this is known as glyceraldehyde 3-phosphate. We made 4 ATP. We used 2 up here. So we have a net gain of 2. That's all we get out of glycolysis. All right, is that net gain of 2. However, we do release hydrogens, and those hydrogens are bound to a coenzyme. Sometimes this is written as NADH2, sometimes it's NADH plus that. Okay, so you might see it either way. This, what's a coenzyme? Anybody know what a coenzyme is? Does it speed up the rate of a reaction by interacting with the substrate? The way a regular enzyme does?
Does it interact with another enzyme?
Usually it's involved in the reaction but not directly. It's not, so here it is bound to our substrate, but it functions, I'm sorry, John?
By changing the binding side of the substrate itself?
Okay, at this point that's not what it's doing. In this case, what it's doing is just being a transport. It's taking the hydrogen from the cytoplasm and transporting it to the mitochondria for the next reaction, instead of waiting for that to happen by chance. All right, so it's functioning as a transport molecule for the hydrogen in the cytoplasm to the mitochondria where we're going to have our next reaction. So this is one of those reasons why electron transport, not the reason why, but this is where we get the rest of our ATP. It's from these hydrogens that are released. So we don't see it directly in glycolysis. We'll see the ATP from these hydrogens. We're going to get 3 molecules of ATP from each of these. So even though we got 2 from this entire process, by the time we finish processing these hydrogen, we'll get 6 more. Okay, of the 38 that we're going to get in the first place. Now, the component of, actually I skipped a step here, being a long day. I'm sorry guys, I thought I was too early and I was. Because I said we weren't going to make any ATP, so let you catch up with me. All right, so the 6-carbon glucose is split into 2 3-carbon glyceraldehydes, and that does not release energy. That was the step that's shown right here. And it's when we take glyceraldehyde and make it into pyruvic acid that we capture this energy. We have 2 of these. And now this step, when we form pyruvic acid, take off the phosphate, now we get the 4 ATPs. And bind the hydrogens to NAD, okay? So this is the energy production state. So one of the things in the summary that I want you to know is what are the products of each of these components. So the products of glycolysis are what I have down here. Pyruvic acid.
Don't you get 2 molecules of pyruvic acid.
Yeah, right here. So pyruvic acid, ATP, and the hydrogen bound to our coenzyme NAD. Plus what else? That I don't have written here.
An extra 2 hydrogen.
That's on here. We have
But
two.
it also says 2 NADH.
Well, that's what's found here.
Oh, okay, I see.
Okay?
Sorry.
Well, there's one other thing that I don't have written here that we always, that we're gonna have. Heat. Because we only capture about 50% of the energy produced. We only capture about 50% of that as ATP, right? The phrase is sometimes used, the rest is lost as heat, and as you know, we don't ever lose energy. It's just it's not utilized by the body for work. You get warm, okay? But it's not utilized by the body to create work. So 50% of the energy released in this reaction is captured by making 4 ATP molecules. The rest of it is lost as heat. So that's the end of glycolysis. I'm going to do just the intermediate step, and then we'll stop with that. So this can take place in the cytoplasm without additional oxygen. All right? It's not a very good way to lose weight. I had a friend once whose husband would go on these exercise binges, and he would get up really early in the morning, go to the gym, and ride the exercise bike as hard as he could for an hour, and wonder why he wasn't losing weight. All right? So for good weight loss, it needs to be aerobic metabolism. In fact, there's some evidence that people can lose more weight. It's never worked for me. Just by breathing more oxygen, taking in deeper breaths. It just seems way too easy.
Yeah.
I'm gonna make my way to weight loss. All right, so we'll come back and review this when we do citric acid cycle on Tuesday, but I want to do the intermediate step. The citric acid cycle takes a 2-carbon molecule and processes it for energy and hydrogen. Here we have a 3-carbon, all right? Pyruvic acid is our 3-carbon molecule. If we do not have oxygen for the mitochondrial reactions to occur, pyruvic acid will be converted to what? Lactic acid. And that is creates what's known as an oxygen debt. It's the amount of oxygen you need to take that lactic acid back to pyruvic acid and through the rest of the citric acid cycle. So the intermediate step is the first time we actually see the formation of carbon dioxide. Because when we went from a 6-carbon molecule to 2 3-carbon molecules, we broke a carbon-carbon bond, but we did not actually release a carbon. Those 3 carbons were still captured as pyruvic acid. So in the intermediate step, if we have oxygen available, this will not occur without oxygen being available, pyruvic acid is converted to a 2-carbon molecule, and we have 2 of those, obviously, because we have 2 pyruvic acids, known as an acetyl group. In addition, we have extra hydrogens, which are bound to our coenzyme. And for the first time, we make the waste product you're already familiar with as carbon dioxide. So each pyruvic acid molecule is going to release 1 molecule of carbon dioxide. Okay? We still have 4 carbons left. We still have more carbon dioxide to make. The acetyl group now is bound to a coenzyme known as coenzyme A. And so we have the molecule acetyl-CoA to transfer those 2 carbons to the mitochondria for citric acid cycle. This doesn't produce any ATP. Okay? We do have hydrogens, that we're going to process in electron transport and get more ATP from. But as an actual step, this does not produce ATP. Okay? Now, this whole process that we're talking about comes under the heading of metabolism. However, it is specifically referred to as catabolism. All right, remember, well, you weren't here. It was in the morning. We had 2 case studies, both dealing with anabolic steroids. So the 2 steps of metabolism are anabolism and catabolism. And when I was in school, the way I kept them straight was catabolism and catastrophe. Okay, things aren't usually built during catastrophes. And so breaking glucose down to its component carbons and oxygen and hydrogen is breaking things apart, and that's glucose catabolism. Now, if you have excess glucose in your body, what does the liver do with that? It binds glucose to glucose and makes starch and glycogen. Glycogen is a form of starch. That would be anabolism. Okay? Or the body taking the carbons and the hydrogens and making, along with nitrogen, making amino acids.
Anabolic steroids.
Exactly. So building things together, anabolism, breaking things down, catabolism. All right? So this process of glycolysis, the lysis or breakdown of glucose, is the catabolic activity. All right, so we'll stop there. I heard that. And on Tuesday, we will review this briefly, continue with citric acid cycle and electron transport, and probably 2 40-minute lectures. We'll finish this and then do the hormones. Okay? Hormones is just really briefly an outline, again, of the effects on the blood levels of amino acids and fatty acids released in those hormones that we covered. That was covered in the PowerPoint for the case study this morning. As soon as those are emailed to me, I'll put them up on D2L. And you guys got it.
For all you guys out there.
For that lecture.
Oh, too late. Booty!
Your quiz on Tuesday.